Hey guys, welcome to your lab on cellular reproduction. That is mitosis and meiosis. Now you've learned in lecture already what the difference is between mitosis and meiosis. The differences, I should say. Just recall that mitosis is reproduction, i.e. cellular division, of all the cells in your body except for the sex cells. So except for the gametes, eggs for females, sperm for males. So we're going to, in this lab, be illustrating cells in mitosis, specifically onion root tip cells, and we're going to be practicing the phases of mitosis and meiosis using Bendy ruse. So you find the Bendy ruse in the little bag called Mitosis Meiosis. And what are Bendy ruse, you ask? Well, they're actually little things for kids, and they usually come in long pieces, and the kids can play with them and paste them to car windows and things like that, but they make excellent models of chromosomes. So let's get started with what you're going to do with the onion root tip cells first of all. So we are going to be breaking out our handy dandy microscope, make sure it has fresh batteries, and I'm going to go through how to focus it pretty quickly because you should already be an expert microscoper back from your microscope lab from before. You're going to want to fetch out of your slide box. Your slide box, this slide right here. It should say onion root tip allium, LS means long section, and I don't know if you can see it on the thing here. You can actually see these little root tips on there. So sometimes if you hold the slide up to the light and look, you'll just be able to make sure you have your onion there. You might want to have your lens paper handy for cleaning your slide and for cleaning the lenses of the microscope. So you just need a small piece. I'm just going to tear that off. I'm going to go ahead and clean off my slide here and clean the lenses of my microscope. Remember, you never want to use paper towels or anything else except for lens paper on your scope because it can scratch the lenses. All right, so turn it on. There's the light. And very gently place the slide here. Now, if you can't remember mitosis and meiosis, just realize that I put some links on the lab handout that has some video tutorials to go over those steps. And of course, you can always view my lecture on mitosis and meiosis in the lecture part of the course. But just a quick review, remember that mitosis involves 4 main steps. So interphase is not a phase of mitosis. That's between the different phases of the cell cycle. So our fingers are going to be the chromosomes, and so in prophase, the chromosomes have condensed because the DNA has wrapped around the little proteins called histones, nuclear envelope has disappeared, centrioles have started to migrate to opposite sides of the cell, and so our fingers are prophase, and they look like this. Okay, so prophase, then they line up down the middle, metaphase, then Anna kicks her boyfriend out, they move to opposite sides of the town, so the chromosomes are migrating to the poles of the cell, that's anaphase. And then as the cell starts to divide, the chromosomes turn back into the stringy chromatin stuff, and a nuclear envelope forms around each set of chromatin, and eventually that cell splits and you have 2 cells, each with identical amounts of genetic material, identical chromosomes, 2 identical daughter cells. So go back and review mitosis and meiosis if you don't recall it. In this tutorial, I'm just going to show you how to do the lab itself. So gently put your slide on the stage under the clips. Now remember that you always want to start on the lowest magnification, so we're going to start at 4x. You may need to adjust your light, so remember you can do that with this. So you can adjust your diaphragm here to adjust the amount of light coming through. And you may have to slide your slide around until you actually see the onion root tip in there. Now, with the onion root tip, you want to go to the area where there's the most number of cells in mitosis. And so that's usually about 1/3 up from the bottom tip of the onion root. I drew here kind of a little bit of a onion root tip, what it might look like under low power. And notice that you're not going to want to be focusing on the very bottom. You're going to want to go up and focus about 1/3 up the way. These cells in here are the ones that are most likely to have the most number of cells in mitosis. But you're going to want to start off on 4x, the lowest, so you won't be able to see much detail of the cells at this point. You'll just see the root tip itself and maybe some little dots, which are actually the nuclei of the cells. So after you get it focused on the lowest objective, you'll just see the outline of the main root. You want to change it over clockwise to the yellow one. That's 10x, so 100 times. Focus it. Here's where you can really try to focus in on that 1/3 from the bottom. And you'll see the dark dots. Those are the nuclei that are in mitosis of some sort. And then switch it to the high power and slight focus. You might need to adjust the light, and you might need to move the slide down. So try to get to an area where you see the most number of cells in mitosis. And so I've included on your lab handout links to websites that will show you example pictures of the different phases of the cells in prophase, metaphase, anaphase, telophase. So you want to find ones that are in all these phases, and the idea is you're going to count 50 cells that are undergoing the different phases of mitosis, and out of those 50, how many are in prophase, how many are in metaphase, how many are in anaphase, and how many are in telophase. Okay, so I've focused my cells under 400 total magnification, the 40x blue ring objective, and I took a picture. Here's what I got. It's a little fuzzy, but okay, so I got a picture on my microscope. This is under 400 total magnification, so the 40x objective, that's the one with the blue ring on it. And this picture didn't come out so good, but just to give you an idea, again, definitely visit the links to the websites I put on the lab handout, because those will show some nice color photographs of onion root tip cells. Let me just show you a couple that I can identify here. You see these ones right here. This is telophase, and how do you know it's telophase? Because you can see that the cells are still small. These are plants, so they produce a cell plate in the middle to divide, as opposed to furrowing like happens with animal cells. But so these are the two new nuclei forming here. So that would be a telophase. These are prophase or early prophase, late prophase. This is probably actually later here because you can see the chromosomes starting to condense. Let's see. Over here, I think this is an early anaphase because you can see these chromatids starting to separate with some spindle fibers in the middle. Let's see if I can find a better example of anaphase in here. Unfortunately, these ones are not a very good example. But when you're looking for anaphase, try to look for something that'll look like this, except you'll actually be able to see the spindle fibers between them and no cell plate. So they'll be further apart, but it'll all be within one cell, and that's anaphase. Over here, this is a metaphase. You can see how the chromosomes are lined up right down the middle of the cell. And in later metaphase, you'll actually see the chromatids will look like these like spindly strings coming out. But you'll see that most of these on here are prophase. So what you need to do for the lab is count 50 of these cells that are undergoing mitosis. Don't count ones like this that are in interphase. Count the ones that are actually in mitosis, and out of those, how many are in prophase, how many are in metaphase, how many are in anaphase, and how many are in telophase. Okay, now the fun part. We get to play with our Bendy ruse. So for mitosis, you're going to want just, you could take 3 colors, you could take 4 colors. Just remember that in a human, the diploid chromosome number is 46. We don't have enough Bendy ruse to illustrate a diploid organism with a diploid number of 46. So we're going to say 4. Okay, so you're going to need 4 colors here. So in my kit, it's orange, red, blue, brown. I'm not sure if it's the same in yours. But you're going to need both copies of each color. So I'm going to bring this screen down, and hopefully you'll be able to see what I'm doing here on the table. Okay, good. All right. So can you guys see that? Okay, so here are my Bendy ruse. And what I'm going to do is first illustrate prophase. So I'm going to go ahead and know that my chromosomes are already duplicated, so they look like X's. Okay, so I'm going to duplicate my chromosomes by taking the two chromatids of each color. And okay, I got my chromatids here. So in prophase, the DNA has condensed. It's condensing, and it's wrapping around little proteins called histones. And it's duplicated, so these look like X's. That means there's duplicate chromatids for each chromosome. So for this particular organism, maybe it's some kind of insect or something, my diploid number is 4. Now remember that in every diploid cell, you got half the number of chromosomes from your mom and half of the chromosomes came from your dad. So in prophase, you'll start to get little centrioles forming, and these are organelles. They'll start appearing, and they're going to start migrating to opposite sides of the cell. they're going to produce little proteins called spindle fibers, which will start to attach to the centers, the centromeres of these chromosomes. So these little spindle fibers are attaching. So I'm illustrating prophase here. Now the next phase is metaphase, and again we're talking about mitosis here. So metaphase, these chromosomes will start to line up down the middle of the cell. And it's important that they line up perfectly, otherwise they will not split evenly, and which is especially a bad thing for meiosis, which we'll talk about in a little bit. Okay, so that is metaphase. Anaphase, Anna kicks her boyfriend out. So now these chromatids are going to start to separate. So we can illustrate that with pulling these chromatids apart, and they start migrating to opposite sides of the cell. Okay, so I'm illustrating that by pulling these bendy ruse apart. So remember, each chromatid by itself is still representing one chromosome. So now I have my two sides of my cell here. So my cell is all around here, but these chromatids have pulled apart and are moving to opposite sides of the cell along these spindle fibers. Now the final phase of mitosis is telophase. So these chromatids are now at each centriole, and they are turning back into that stringy stuff called chromatin. And they're actually forming a new nuclear envelope around each set of chromatin, and eventually that cell is going to pinch, and boop, now you have two identical daughter cells. And remember, because each chromatid represents a chromosome, if you started with 4 chromosomes in the mother cell, each of the daughter cells also now has 4 chromosomes. And so the diploid number is still 4, it's been conserved, and each daughter cell is genetically identical to the mother cell and identical to each other. Okay, so now you have 2 identical cells. So I'm just going to go through that process again. Prophase, the DNA has duplicated. It's condensed, wrapped around histone proteins, and so we get things that look like X's here. And of course, in real life, your chromosomes don't come in different colors. They're all the same color, but we're just going to illustrate that. Okay, so they've condensed. Now, metaphase, they are going to line up perfectly down the middle of the cell. Anaphase, the chromatids separate and go to opposite poles with their respective centrioles. The spindle fibers are pulling them across. And telophase, the DNA gets stringy again, it unwinds from the histones, turns back into chromatin, and you end up with the same number of chromosomes in each cell as you had in the mother cell. The cell pinches if it's an animal cell, or if it's a plant cell, it forms a cell plate in the middle, which will grow into the cell wall, which is what you see in your microscope. So in a human, the diploid number is 46, and so each daughter cell would have 46 chromosomes. These could become your skin cells, your liver cells, your heart cells, whatever cell, except for your sex cells. If the diploid number was 8, each cell would have 8. But they're all genetically identical to each other because you don't want a liver cell growing where your skin should be. Okay, so that's mitosis. You're going to draw out these phases. You're going to just practice this as much as you need to really learn the process. But now we're going to learn about meiosis. Let me pull this up here for a second again. So meiosis is the production of sperm or eggs. So the production of sperm is called spermatogenesis, and the production of eggs is called oogenesis. To illustrate this, get out your duplicated bendy ruse again, but this time you're going to need pairs of chromosomes. So in this case, we're going to make an organism that has a diploid number of 4, and so remember, in meiosis, you're producing your eggs or your sperm, and you want the eggs or sperm to have half the number of chromosomes that the parent cell started with. Now, why is that? Well, because when egg and sperm come together to make a baby, you want to make sure that you're not having too many genes. So you want half the number in the sperm plus half the number in the egg will give you the whole amount in the baby that you want. So if this was humans, we'd start out with a diploid cell that has 46 chromosomes, meaning the egg and sperm each have 23. But because we don't have that many bendy ruse, we're going to pretend that we have an organism that has a diploid number of 4, which means the eggs or sperm it produces will have 2. But unlike mitosis, these sperm and eggs are going to be genetically different from each other and from the mother cell, which is why if you look around, you'll see everybody looks different. You even look different than your sisters or brothers unless you're an identical twin, and it's because of the things that happen in meiosis. So we're going to illustrate this In this case, you've got to remember that you got half your chromosomes from your mom and half from your dad. And so, in the case of humans, chromosome number 5 from mom matches up with number 5 from dad, 7 from mom matches up with 7 from dad, et cetera, et cetera. And so those same kinds of chromosomes are called homologous chromosomes. Okay, so watch what I'm going to do here. And again, watch the video tutorials that I've put associated with this lab, and watch my lecture video explaining how meiosis works. This is just practice. Okay, so back down here. So here's what I got here. And remember that meiosis occurs in 2 main phases, meiosis 1 and meiosis 2. And in meiosis 1, we're going to generate genetic diversity. So prophase 1, so remember there's 2 sets. Prophase 1, kind of like before, the chromosomes are forming, the DNA is condensing by wrapping around histone proteins. But towards the end of prophase, these homologous chromosomes start to pair up down the middle of the cell. And that's completed in metaphase 1. So if this was chromosome number 1 from mom, it would pair up with number 1 from dad. This is number 4 from mom, this would pair up with number 4 from dad. But in our case, we only have 2 pairs of chromosomes. So 1 from mom, 1 from dad, 2 from mom, 2 from dad. Or they could have assorted this way. Maybe dad's was on this side and mom's was on this side, and vice versa. Could have gone any different combination within these pairs, and that is the law of independent assortment that Mendel came up with. The idea that chromosomes assort independently in late prophase 1 or early metaphase 1. And that helps with the 50-50 chance that you'll inherit more of your mom's or your dad's chromosomes. So you might notice that maybe you look more like one parent and your sibling looks more like the other because you literally express more genes from the other parent. So in metaphase 1, they finish pairing up, homologous chromosomes pair up, but then the other thing that happens is that the chromatids can actually touch each other. So each one of these is a chromatid, and if they touch, they actually will swap DNA in that location. And so we can illustrate the swapping, called crossing over or recombination, by switching the colors of the chromatids. Okay, so now I'm going to make an X that's orange and blue here, and this one will be red and brown, and this one will be red and brown. I hope this is showing up on the camera. Okay, so this illustrates that crossing over happened by these chromatids actually touching. So you actually exchanged material, genetic material, between the genetic material from the 2 parents. Okay, and now, so that was metaphase 1. We had independent assortment and crossing over. Those are the 2 things that lead to genetic diversity in organisms. Okay, so anaphase 1, instead of having the chromatids separate like happened in mitosis, the entire duplicated chromosomes will separate. So we can illustrate that like that. So already you can see that what's going to become each of these 2 cells are genetically different than what we started with because they have genes from both parents. Okay, so anaphase 1, telophase 1, same kind of thing. They're going to condense. So by the end of meiosis 1, at the end of telophase 1 of meiosis 1, we have 2 daughter cells, but they're genetically different from each other and from the mother cell because of crossing over and independent assortment. And then in meiosis 2, we're going to start off with prophase 2. And already you'll notice that something else is different that happened by the end of meiosis 1. Each of these new cells is haploid. That means it has half the number of chromosomes than the mother cell. We started off in this case with 4 chromosomes in the cell. Now each of these daughter cells only has 2, and they're genetically different from each other and from the mother cell. So prophase 2, again, the DNA is condensed, the nuclear envelope disappears, all that kind of good stuff. And now meiosis 2 is going to proceed very much like mitosis. So by the end of prophase 2 or the start of metaphase 2, these chromosomes are going to line up perfectly down the center of the cell, of each of these 2 cells. Now this is really important that they line up perfectly, because if they don't, and the same thing could have happened in metaphase 1, if they don't, then when they go to split in anaphase, in this case anaphase 2, you can have daughter cells that end up with too little or too many chromosomes, and that causes a genetic disorder. So for example, this is how Down syndrome happens, where you end up with 47 chromosomes instead of 46 because you got an extra copy of chromosome number 21 due to these not splitting evenly, and that process is called non-disjunction. So this is metaphase 2. Anaphase 2, just like mitosis, the chromatids are now separating. And same thing's going to happen over here. Chromatids separate. And telophase 2, you now, oops, my genes are jumping. You now are going to end up with 4 daughter cells. Each is going to be haploid, so it'll have half the number of chromosomes as the mother cell. Make sure they're all in the camera there. So this represents 1 cell, 2 cells, 3 cells, 4 cells. They're all haploid. They have half the number of chromosomes as what you started with, and they're all genetically different from each other and from the mother cell. And each of these, if this is going to be a guy, these will each become a sperm cell. So they'll grow a little flagellum, little tail, and all the other parts of a sperm cell. And you potentially could have 4 viable sperm cells from that one mother cell, and they'll all be genetically different from each other. So for every 1 mother cell that's diploid, you can produce 4 haploid, genetically distinct sperm cells, all capable of producing a baby. However, women are a little bit different. If she's a girl, then she's going to produce all the eggs she's ever going to have as an embryo, but she won't complete meiosis 2 until fertilization. However, here's the sad part about being a girl. We waste a lot of energy because out of these 4 daughter cells that get produced, only 1 can become a good egg. The rest are going to be little things called polar bodies, and eventually they will disintegrate and go away. And so you end up actually with 1 good fertilizable egg cell for every 1 mother cell. So she does produce 4 daughter cells, just like the sperm, but only 1 is going to be viable. The others disintegrate. So this is called the cost of meiosis. And in animal behavior, they think this actually could be a reason why females invest more in their offspring and therefore are the choosier of the 2 sexes. Anyway, so practice meiosis, practice mitosis with your Bendy ruse. These are just a learning tutorial, but definitely find your onion cells in mitosis in the different phases and figure out how much time they spend in each phase. And let me know if you have any questions. Don't forget to take a picture of your cells in mitosis, and don't forget to take a picture of your Bendy ruse. All right, see you on the discussion board.